Hello, everyone. Welcome to the National Atomic Testing Museum's conversation with Dr. Paul Dabisch of the National Biodefense Analysis and Countermeasure Center. Before we begin, I would like to share with all of you that we have an upcoming exhibit in partnership with the Department of Homeland Security's Science and Technology Directorate that will be highlighting the current COVID-19 research being done at the NBACC. It's now my pleasure to introduce Dr. Paul Dabisch. Now, Dr. Paul Dabisch is a senior principal investigator and the aerobiology team lead at the National Biodefense Analysis and Countermeasure Center at Fort Detrick, Maryland. And Dr. Paul Dabisch's research focuses on factors affecting the aerosol transmission in infectious disease. And he has recently published the results of several studies examining the survival of SARS-CoV-2, the virus responsible for COVID-19, in aerosols under different environmental conditions. He's an active member of the American Association of Aerosol Research, where he has recently served as the chairperson of the Bioaerosols Working Group, as well as organized several symposia, including a special session entitled The Role of Aerosol Science in Understanding the Spread and Control of COVID-19 for the Society's 2020 virtual conference. Dr. Dabisch holds adjunct appointments in the School of Systems Biology at George Mason University and the Department of Biology at Hood College. He received a BS in Crop Sciences from the University of Illinois at Urbana-Champaign and a PhD in pharmacology from the Tulane University School of Medicine. Dr. Dabisch, thank you so much for joining us today.
Well, thanks, Joe. It's a pleasure to be here.
Well, thank you again for joining us and agreeing to be here today. So we'll jump right into the questions. Now, for those of you who are watching, we will be taking questions in the chat and Q&A boxes. So if you do have any questions for Dr. Dabisch, we'll be taking those at the end. But if you'd like to write your questions as we talk with Dr. Dabisch, you're more than welcome to do that. So, Dr. Dabisch, some people watching this may not know much about the National Biodefense Analysis and Countermeasure Center. Can you please give us a brief background on the center? For instance, when was it first established and for what purpose?
Sure. So it's the National Biodefense Analysis and Countermeasure Center, or that's sort of a mouthful. We say NBACC for short. So that is the Department of Homeland Security's lab up at Fort Detrick, Maryland. And so that's about, an hour to 3 hours, depending on traffic, northwest of Washington, D.C. And so it's a biocontainment lab, which means that we work with infectious organisms that cause disease. And so we can work at that. We can work with those organisms at different levels. And so you'll hear it referred to as biosafety level 4, for example. So that's the highest level. So that's the level you would use to work with something like Ebola, for example. And that's what you see in the movie. So in Contagion or Outbreak, you see people wearing these suits that look like space suits. And so that's biosafety level 4. So SARS coronavirus 2, which is the virus that causes COVID-19, requires us to work at what's called biosafety level 3. So this is not as stringent as biosafety level 4. And so when you come into the lab, you're still changing out of your clothes, you're putting on scrubs, you're wearing a positive pressure respirator over your head and gloves and things like that. And all the work is still done in specialized cabinets to contain the virus. And then you're showering to go back out, but we're not wearing these big positive pressure suits like you see in the movies. And so the lab was established in the early 2000s after the Amerithrax attacks. The current facility that we're in went active in 2011, and so that's the lab on Fort Detrick. And there's 2 centers at the lab. So the first is called the National Bioforensics Analysis Center, and they support law enforcement and the FBI. So for biocrimes type things. So if a letter containing ricin was mailed to somebody, the letter would go there, and they would do all the analysis on it to determine whatever they needed to know about it. I sit in what's called the National Biological Threat Characterization Center. And so the point of our group is to perform laboratory studies to understand the threat posed by bioterrorism normally. And so that's the focus of our research. And so the results of that research then go on to inform things like, how many drugs do we need to put in the national stockpile in order to protect against a bioterrorism event, things like that. Occasionally, when there's a public health emergency, we'll get asked to focus on that. So in 2014 to 2016, there was a large Ebola outbreak in West Africa. We got asked to look at different disinfectants for that and how well they worked at killing the virus on different surfaces. And so with COVID-19, we've been asked to look at similar things, and I'll elaborate on that a little bit as we get through the questions, I think. And then I've been there since 2011. So when the new building went online, I started there, and I lead what's called the Aerobiology group. And so for people who might not be familiar with what that means, is we study transmission of disease through the air in aerosols. So little tiny particles that are not really visible to the naked eye that could transmit disease.
Perfect. Thank you for covering that for us. It's a great introduction to the work you do, and like you said, with aerosol and what that actually means for infectious diseases. So speaking of COVID-19, starting in this February, the NBACC has been, or NBACC, as you say, has been researching COVID-19, including the survivability of the virus on commonly used surfaces and in the air. Now, what are some of the key findings of this research?
Yeah, so that's what we've been working on, or at least initially what we worked on. And so I guess to put that in context for a second first, so COVID-19 is a respiratory disease, which means if you go and look at people who have it, you can find the virus in their lungs, you can find it in their throat, you can find it in their mouth. And so in order to infect another person, that material from the respiratory tract, as we call it, would need to somehow get transmitted to that person. And so there's a number of ways that can happen. I think maybe the most obvious way that people would be familiar with is coughing. So obviously when you cough, you expel a large amount of material out of your respiratory tract. And so some of this is very large particles, things you can see, and those are going to fall down to a surface. And if someone else were to come along and touch that surface and get it on their hand and then rub their eye or put their hand in their mouth, they could potentially become infected that way. And so that's what we would call fomite transmission. Also, when you cough, a lot of the particles that are generated are actually very, very small. And so they're not visible to the naked eye, and they can remain aloft in the air for a long period of time. And they can actually move quite a distance. And so if someone further away were to inhale one of those, they can become infected that way. And I mentioned coughing, but you get the same sort of phenomenon occurring in terms of you're expelling particles when you talk or you sing. So it's not as many, but you have the same thing where you have some large ones, some small ones, some will fall to the ground, some will stay in the air. And then even when you're just sitting here breathing right now, you're generating very small particles that are coming out of your nose or your mouth that can hang around in the air for a substantial period of time and potentially infect people. And so some of the initial questions we wanted to answer were revolving around transmission. And so if somebody did expel these particles and they landed on a surface, how long would they actually remain infectious on that surface? And so did we have to decon it right away, or would they die really quickly on the surface? So we wanted to answer that question. We also wanted to know if somebody was breathing out these really small aerosol particles, how long they would actually last in the air as well. And so to do these studies, conceptually, they're pretty straightforward. Execution is obviously very logistically challenging in the environment we're working in, but conceptually they're pretty straightforward. So for a surface study, for example, you're going to take a droplet of the virus, we're going to put it on a surface, and we're just going to let it sit there. And then at some point after that, we're going to look and see how much virus is still remaining. We know how much we put on the surface, for example. And if we do this for a bunch of different times, we can actually estimate the rate at which the virus is dying on that surface. So that's pretty straightforward. We're sort of uniquely positioned at the lab in that we have chambers that allow us to control the temperature and the relative humidity around that surface, as well as illuminate that surface with simulated sunlight. So this would be sunlight that's representative of what you have outdoors during the summer, for example, or during the winter. We can change the level. And so what we looked at was the effect of these different factors, so temperature, humidity, sunlight, on how long the virus survives on surfaces. And so what we found was that all three of those factors actually affect how long it remains infectious on a surface. So for example, in an office environment that's climate controlled, or an air conditioned house, or a movie theater, or something like that, if you had virus on surface, it's going to last. The majority of that virus is going to remain infectious for tens of hours, maybe days even. And so in that case, you have to worry about someone contaminating the surface and it remaining there for a long period of time. If you take that same surface and you put it out in the sun, for example, what we found was 90% of that virus is actually killed in about 10 to 15 minutes, depending on the level of the sunlight that is actually present there. And so that has implications for, in the real world, for things like, what shopping cart am I going to take when I go to the store? So like over the summer, if I went to the store, I looked for the shopping cart that was in the cart return, in the back of the return, that had been sitting out in the sun for a while, instead of grabbing the one that was inside the store. We also found the same thing with temperature. So as the temperature went up, the virus survived less well on the surface. So the rate at which it died went up. And so, under indoor conditions, it lasts a long time on a surface. So climate controlled conditions, air conditioning, things like that. When you get up to 100 degrees Fahrenheit, instead of tens of hours, you're down to, it lasts a couple of hours before the majority of it's inactivated. When you get up to about 130 degrees Fahrenheit, which sounds a little unreasonable, but now you're losing 90% of that virus in about a half hour or so. And so 130, like I said, is a little unreasonable, but keep in mind that in the summer, if you parked your car out in the parking lot, that's about the temperature your car gets to. And so in terms of mitigating, or decontaminating surfaces, parking your car in the sun in the summer and leaving it sit there while you're at work, the risk of being exposed to virus that might have been on a surface in that car is much less after a couple of hours. And so just another example of how the data we've generated can be applied to the real world. So as it relates to aerosols, then you see a lot of the same trends. And so the way we do these studies are similar, but instead of putting a droplet of the virus on the surface, we're actually generating an aerosol into one of these chambers. And then we take samples of the air over time, and then we look at how fast the virus is dying. And so indoors, we found the same thing I mentioned for droplets, is that the virus is very stable in the air indoors. It'll last hours before the majority of the virus is killed. And so in an indoor environment, then, if you're trying to mitigate the spread of virus, you're not going to rely on the virus just dying, because it doesn't die, it hangs out. So you'll do things like increase ventilation, or have people wear masks to stop exhalation of the particles, or space people out so that the virus gets diluted and maybe you're not inhaling as much of it. Outdoors, again, we saw the same thing we saw with the droplets. So if the temperature goes up, if there's sunlight present, the virus dies much faster in aerosol particles. And so we looked at a range of conditions, but if you got up to 100 degrees Fahrenheit and high intensity sunlight, so like summertime sunlight, 90% of that virus would be killed in about 2 minutes instead of hours. And so that's substantially faster. It sounds really fast, but also keep in mind that, air and particles entrained in that air can actually move a good distance in 2 minutes. And so in terms of the impact on that for risk of transmission, we're still working on that a little bit. There's a few other things we need to know in order to really assess that. And specifically, that is, if I have an infected person, how much infectious virus are they actually breathing out or coughing out or coming out when they speak? And then the second thing we really need to understand better is someone who's uninfected and near that person, how much of what's exhaled or spit out do they need to be exposed to actually get them sick? And so if we had a better understanding of those things, and I think we'll come to that a little bit later in the discussion, then we could better estimate, the effect of these environmental factors on the risk of transmission.
Well, that's interesting. Thank you, Dr. Dabisch, for sharing with us what you've learned and also walking us through the process of how you discover these things. Now, moving on to the work you do, of course, you went through all of the things you've learned, but how has your work at NBACC changed since researching COVID-19 became a priority for the Department of Homeland Security? Has it impacted you in any ways in terms of what you do day to day?
Yeah, so as I mentioned sort of in my first answer, our normal focus is bioterrorism. And so since this pandemic started, February, March, I think March 11th was when it was officially declared a pandemic by the World Health Organization, our focus has been almost exclusively on studies to better understand the transmission of COVID-19 and SARS-CoV-2, that's the virus. With the intent of using that data to develop mitigation strategies then to help protect, DHS personnel. So we are a DHS lab, so that's a lot of the focus, but also knowing that a lot of what we find can be applied to the general public and help keep the public safe as well. And so yeah, when that all started, the day-to-day tempo changed at the lab dramatically. So we were suddenly working, had people in the labs 7 days a week, up to 12 hours a day on every day, trying to get these studies done because they were a high priority. And so that became very challenging, getting used to working at that tempo. So we're normally not anywhere near that sort of tempo, right? And that becomes extremely challenging since a lot of things in this area at least were shutting down, and so a lot of people had kids, and so you had to deal with childcare and balancing being in the lab for 12 hours. And so yeah, that became a very challenging time for a lot of people.
Well, thank you. It's interesting to hear, how it did impact everyone, just like it did with, all of us around the world. So now, many individuals, speaking of how it impacted people, while many individuals were able to do their jobs from home during the shutdown, a large number of people were considered essential workers, such as first responders, hospital employees, and those in the service industry. Was special attention placed on the experiences of these people when research first began?
Yeah, that's an interesting question. So DHS, I mean, among the essential employees that you talk about, DHS has a large number of essential employees that are employed. I have friends in Customs and Border Protection who haven't had a break during this whole thing because what they do, they can't just stay home because they're manning the border and doing other essential functions. And so when we started these studies, I mean, yeah, they were explicitly designed to provide data that could be used to protect not only DHS personnel, but the general public as well. But the focus was DHS personnel. And so I talked about the studies we did to see how long it persists in the environment, the virus, but we've also done a number of studies more recently, I mean, back to March, but still ongoing, looking at how well different common household disinfectants and other things will work on killing the virus on various surfaces. And so these studies are done, similar to what I talked about before, where you're putting a drop of virus on the surface and then you're covering it with a disinfectant, and then you're waiting some period of time and looking to see how much virus is still infectious. And so what we found with that is, in terms of household items, bleach, isopropyl alcohol, so that's the active ingredient in Purell. Both of those work very well to kill this virus. Hydrogen peroxide, something also pretty common in the household, doesn't work so well maybe. So that's interesting, and we'll have that data published in pretty soon.
Well, thank you. Now, were there any new tools, where you're obviously talking about how you looked at essential workers and their experience, of course. Were there any new tools or equipment that were developed in conjunction with or as a result of the research?
Yeah, so I mentioned all of the work looking at how long it persists in the environment. And so, yeah, if you take all of that data, you can build a mathematical model associated with that, which then allows you to go and put in, any temperature, any humidity, any level of sunlight, and actually estimate how long the virus would be expected to survive. And so we did that as part of the work we've done, and that's published in some of our peer-reviewed papers. But DHS took that information and used it to build an online tool that is on the DHS S&T website. So I think if you just Google DHS S&T and COVID, you'll find it pretty easily. But what that tool allows somebody to do then, and it's available to the general public, is to go to the website and then you can put in any combination of conditions you want. So a temperature, humidity, and it uses UV index, which you can get off your weather app. And then you can estimate how long the virus would be expected to survive under those conditions, and you can see how the different factors matter.
Thank you very much. That's great to hear. So now, has the research being done at the center impacted the implementation of personal protective equipment or PPE and our understanding of how they should be cleaned or decontaminated?
Yeah, so a lot of the data I talked about already in terms of how long it lasts. I mean, it tells us under what conditions it's stable. And so that can inform development of guidelines to tell us what PPE you should wear and when. And so like in a clinical setting, for example, and things like that. And obviously the work with disinfectants obviously has implications for telling us which cleaning products should be used under different circumstances. DHS S&T's also done a little bit of work, I guess more directly to your question, looking at low-cost methods to sort of sterilize or decontaminate PPE, because some PPE is in short supply still, so that it can be reused. So that's not work that's being done at the lab, that's being done elsewhere, but it is a DHS S&T effort. And so I can't really speak to the details because it's not something that the lab is explicitly doing, but I know if you go to the DHS S&T website and look for that, there's information there if someone was interested in finding out more about what they're doing there.
We'll certainly appreciate you sharing that and letting us know where our audience and us here at the museum, where we can go to discover more about that. So thank you for, again, sharing that. Now, speaking of other parts of S&T, is the NBACC working with any other national laboratories in the US or around the world? Is there any of those partnerships extending beyond the walls of NBACC?
Yeah, so we collaborate pretty extensively across the US government and also across academia, so universities and things like that. So within the US government, we have a project ongoing with Nist, looking at UV inactivation of the virus. So Nist is the National Institute of Standards and Technology. It's part of the Department of Commerce. We've also worked pretty extensively with NIAID. So that's the National Institute of Allergy and Infectious Diseases. It's part of the National Institutes of Health. Outside of the government, we have a project ongoing right now with actually the University of Nebraska Medical Center, a project they're doing. They're looking for the presence of the virus in different patient rooms or clinical settings. And so they're taking samples of surfaces and samples of the air, trying to understand how contaminated these environments get when you have patients with COVID present in there. So we're collaborating with them on that. And then outside the US, we talk to a number of groups outside. So for example, in the United Kingdom, our counterparts over there are our lab called the Defense Science and Technology Laboratory, DSTL. And so we work very frequently with them. And so in terms of COVID, that's been more just data exchange and talking about the different experiments we're planning so that we can make them either complementary or make sure we're not doing exactly the same thing, so we can provide the most data possible. But yeah, those are some of the examples.
Well, it's good to hear. It's good to hear that there's an exchange of that information, even if it's not directly working together on something specific. It's great to hear that there's that exchange. So that's certainly a big part of working together in 2020. Now, in addition to that, are there any updates? Speaking of the work you've been doing, we've talked a lot about what you discovered since March. But are there any recent updates you can share with us in terms of the research you and the team are doing?
Yeah, so what are we doing now? So as I mentioned, I guess a little bit earlier, so there were two big questions I said that we didn't have a lot of information on. So the first was, if there is an infected person present, how much virus are they actually emitting from their respiratory tract? So and is it infectious? And then the second question was, how much of that virus does somebody who's uninfected need to be exposed to get them sick, essentially, right? So there are some data out in the literature, but I would say we don't have a great understanding of either of those pieces right now. And so we have a collaborative project that's just kicking off in 2 weeks, actually, in collaboration with NIAID, where we're going to look at both of those questions. And so obviously this can't be done in humans at the lab. So this is being done in monkeys, actually, but that will hopefully give us some idea as to how much is expelled and how much it takes to get somebody sick. The other thing we're doing right now is we're doing some additional work with TSA. So TSA is a DHS component. It's the Transportation and Safety Administration. And so that's who runs all the checkpoints at the airport. They have questions about different disinfectants that they want to use to disinfect different things at the checkpoints or on airplanes and things like that, and different surfaces that are present in those environments and whether those matter. And so we're working with them right now to look at a range of different disinfectants and how well they work on different surfaces. And they'll use that information then to ensure that travelers are safe when they're traveling. And then the last thing I guess we have going on now that I'll mention is this collaboration I sort of mentioned with Nist, so that group from the Department of Commerce. And so one of their missions is to actually evaluate commercial products that come out on the market. And so a lot of what has come out are, you may see them at the store. I saw a couple at Home Depot the other day, these UV disinfecting lamps and things like that. And so part of their mission is to be able to assess whether these things actually work. And so we're working with them to build a test system right now at the lab that will allow us then to look at, different products or the UV wavelengths of light associated with those products and how well they actually kill the virus. And so we'll provide real data on that, and they can use that then to say, this product would work, this one doesn't. And then from the DHS perspective, we can use that to say, okay, maybe we should develop products, based on this UV wavelength that we could use to decon things at a TSA checkpoint or on an airplane or things like that. So yeah, that's a little bit of what we have going on now, but still very COVID focused.
Great. Well, we certainly appreciate all the hard work and the great work you're continuing to do. And it's interesting to also hear about those upcoming collaborative efforts with the different agencies within DHS and out of DHS. So that's fantastic. Now, you already spoke a little bit about this, but what are some additional resources you can recommend for those looking for more information about the Science and Technology Directorate, NBACC, and your specific research into COVID-19?
Yeah, so like I mentioned, if you Google DHS S&T and put COVID on there, and you don't even have to put the COVID on there, you'll find DHS S&T's website, and you can scroll through there and find information on a lot of what's going on. And so that's a good resource. In terms of the technical information, if there's any more technical folks out there interested in finding the papers, I mean, you can go to Google Scholar or the National Library of Medicine and Google or enter my last name in COVID, and you'll find the papers we've published on the topic so far, and that'll have a little bit more of the technical data in it, and it'll be a little more technical, so maybe not for everybody, but that's where maybe you would go find that information.
Perfect, thank you. Now we're once again opening, first off, thank you so much, Dr. Dabisch, for going through all of those details for us. Great information. Certainly a lot of people looking to hear what is actually being done in terms of research. And so knowing that there's this big center within DHS working on this, it's great to hear. And also, of course, learning about all of your important findings. Now we are going to open up questions, of course. Chat box is available to you, as is the Q&A box. So if you have any questions for Dr. Dabisch, please feel free to write in. I think we do have one of those right now. Let me see here. So how exposure, concentration / viral load and length of exposure time affect the course of disease? So how does the exposure, concentration, viral load, and length of exposure time affect the course of the disease?
Yeah, that's an interesting question. And so we don't have a good idea for SARS-CoV-2, obviously. As I said, one of the big gaps is, how much virus you actually need to be exposed to cause disease. And so once we have a good idea of that, we can start looking at some of these other questions like exposure duration. What we do know from other studies in the literature, though, for microorganisms at least, so that the duration over which you're exposed, as long as it's less than, say, 12 hours or something like that, doesn't seem to matter, because keep in mind that when you're exposed to this, that disease takes, several days to get going and to manifest. And so if the exposure period is short, and when I say short, I mean less than 12 hours, it doesn't seem to affect the disease presentation or the time course. And so there's some older data from the 60s where they were looking at this for anthrax, for example, and that's what they found. If you look at it for chemicals, so if I was exposed to a chemical like in the household, something toxic, the exposure duration matters much more, but that's because the toxic effect from that is happening almost instantaneously. And so if I'm exposed to it all at once, I will have a very different response than if I was exposed to a really low level over a long period of time. So those are two different situations. But for biologicals or microorganisms that you would breathe in, it doesn't. I wouldn't expect it to have a huge effect on what you see in terms of the presentation of the disease several days later. But again, that's an unknown still with COVID, honestly.
Certainly. Thank you so much for answering that question. That makes a lot of sense. Something we're still learning as we go, of course. So one question is, are there any, and I think you touched on this a little bit, but are there any known materials or chemicals that repel the virus or at least keep it at bay?
Yeah, repel. I don't know. Exactly what the questioner means, but like I said, there are things we know that you can use to disinfect surfaces, and so if you did have someone coughing at a table and at a restaurant, and you saw stuff land on the surface or after they left the table, we know if you wipe that down with a dilute bleach solution or isopropyl alcohol or something like that, that will kill the virus and clean that surface. And so, I mean, not exactly sure what was meant by repel, but we do know things that can stop it. We know masks will protect both the wearer and someone else out in the room who may be exposed to the virus. And so even if you're wearing it very poorly, there's data that show that it stops those exhaled particles I talked about. And so, or at least redirects them, and so that they're not moving out at a great velocity from your mouth. And so one of the things that happens if you're talking or even breathing or coughing is that air is actually being propelled out of your mouth at a pretty substantial velocity in some cases. And so that allows those particles to get pushed really far away from the source, for example. And wearing a mask, there's data out there that have been published shows that redirects that velocity. So it slows it down right away, and sort of, instead of going straight out from your face, it'll push it off to the side. And so the exposure for people around you is a lot less. And if it's a very efficient mask, it'll also stop the particles. But even if you were wearing it poorly, it'll still redirect some of that flow.
Well, that's interesting to hear about the masks and how even if worn poorly, it still has an effect on the trajectory and also protecting both the wearer and people around them. So that's really useful data. Thank you. And so referencing the one about repel, they just mean specifically repelling air droplets. I think that's what they were getting at with anything that repel the droplets themselves. But I think you answered that.
Yeah, I mean, larger droplets are going to fall to a surface, and there really isn't anything that's going to stop that when you get to particles that are visible. I mean, gravity's pulling it down, and it's wet. There's not much you can do to stop it from hitting something. The smaller particles, they're following the air streams, and so you can play around with airflow and things like that to shunt particles away from certain areas. So there's a lot of simulations going on right now as part of some of the conferences we've been involved with, looking at how to change airflow in a room in order to minimize exposure of the people in the room. So yeah, there's some work going on there, but yeah, airflow is really the one way to sort of repel small particles.
Great, thank you for revisiting that one question. I appreciate it. So another great question is, how do different materials such as stainless steel, fabrics, ceramics, plastics, etc., relate to the survival time? Does the actual different material make any sort of difference in terms of how long it can survive?
Yeah, that's a good question. So I didn't really dwell on it, but as part of our studies, we did look at different surfaces. So we looked at stainless steel, so that's common in clinical settings. We also looked at a type of plastic that's very common in cell phones and keyboards. And what we found was there really is no difference in how long it survives on something that's non-porous like that. So when I say non-porous, I mean that the liquid doesn't soak into it, right? And that's been known for a while for other things that people have looked at. So people looked at, during the original SARS outbreak back in the early 2000s, people looked at a whole range of different surfaces and found the same thing. If the virus is on a non-porous surface, it tends to survive the same. The one exception there was there are some data out there for certain metals. So copper, for example, if there's copper present, it seems to actually inactivate the virus on that surface a little bit faster. In terms of non-porous things, so you said fabrics and things, that's a little trickier to evaluate, because if I put a droplet on fabric, it obviously wicks into the fabric depending on what it is. And so part of developing that sort of study is to come up with a way to recover all of that back out of the fabric, so that I can tell you, after I leave it sit there for a little bit of time, how much is still infectious. And so a lot of that actually gets sort of sequestered into the fabric and isn't really recoverable afterwards. And so there are some studies out there that have said, on cotton, it seems to go away really fast, but some of those studies are actually lacking what we would call the proper controls. And so where they have shown that they can actually get everything back out. So they might not be able to recover it, but that could just be a function of the fact that, it's kind of stuck in the fabric and the method they're using to try to get it back out isn't working. Yeah.
Well, great. Thank you for breaking that down for us in terms of the materials, and also sharing the fact that, there's data out there, but it also depends on, obviously working within the confines of having, more control over that. So that's good to know. Now, one that's, we have another question, and it's related more towards locals in Las Vegas or those visiting. Temperature changes in the fall and winter, are they more likely to cause concern even in Las Vegas, where, we don't get the harsh winters of, say, the East and the Midwest? Is that something that's still a concern, though? Because it does significantly drop, compared to the summer.
Yeah. So, I'll caveat this with saying, like I mentioned, we don't understand exactly yet the role that environment plays in disease transmission. And so there's a lot of factors there. And so what we looked at was, does the environment changing itself actually inactivate the virus? And the answer there is yes. Now, whether that substantially changes the risk as the environment changes, we don't know, because there's a lot of other things at play here. For example, in the winter, people tend to spend a lot more of their time indoors. And so now you're moving yourself away from an environment that maybe has sunlight, moving yourself away from an environment that may have higher temperatures. Well, not in the winter, I guess, but and where people are in closer proximity to each other. So that can affect transmission as well. And so there's this interplay of lots and lots of different factors that we don't quite have a great handle on yet. And so I think as we get more information on how much virus infected people shed and how much it takes to make somebody sick, we'll see more of these simulations and modeling coming out that can start to answer those questions. But there have been some epidemiological studies published where they looked at, broad environmental conditions and temperature and UV trends across areas that have implicated, temperature, humidity, and UV in the spread. But those are sort of very broad conclusions based on an entire region. So I guess the answer is we don't really know, but it's possible.
Well, thank you for being honest about that. So yeah, it makes a lot of sense, as we're learning and as we're getting into, having been in March in the spring, you're getting into a whole new kind of season. So it would be interesting to see what you learn as you go. So thank you again for answering that. We do have time for a couple more questions if anybody would like to ask any additional questions for Dr. Dabisch. I do have a question. It's something that I've heard people ask a lot recently. You had mentioned that masks are effective for both the wearer and people around them. I have heard people counter, well, if they're effective, then why do we still need to have that that distance between one another, that social distancing? Can you speak a little bit more about why it's important to have both of those and not just an either or situation?
Yeah, sure. So, in terms of distancing, that 6 feet rule has been out there. But, as I talked about at the beginning, if you expel especially really small aerosol particles while you're talking or you're singing or you're breathing or even coughing from the respiratory tract, they can go a lot further than 6 feet. But the point to distancing is that the further you get away from that source, the more dilute that aerosol becomes. So it's moving away and it's getting diluted with other air. And so the concentration of the infectious virus in that air is less. So if you breathe that in, you're less likely to get infected. And so, like I said, we don't know exactly how much you need to breathe in yet, but as you move away from the source, it'll be more dilute and you'll be less likely to breathe that in and become infected. So that's the rationale behind distancing. And then masks is sort of, trying to control the same thing, but in a different way. And so it's trying to block emission of the particles right at the source. So I put it over my mouth and it prevents the particles from getting into the air in the first place. And so there's again, a lower concentration of particles out in the environment that people can breathe in and get sick, right? So they're attempting to accomplish the same thing. And so since they are different mechanisms of accomplishing the same thing, putting them together compounds that effect, and so you'll be more effective that way. So could you do one and have some effect? Yes, you could. But doing both of them together has a much greater effect than either of them individually.
That makes perfect sense to me. Absolutely. Yeah. The more you do, the more likely you are to stay safe, of course. So like I said, we have a couple more questions. So one is about the terminology that you use. Now, is your use of the term deactivated synonymous with killing the virus, or does it mean something else in terms of relating to the virus itself?
Yeah, I'll say inactivate a lot because, I don't know, there's debates about this, but, a virus isn't technically alive. It's not a metabolically active thing. It's a little piece of RNA inside of a little membrane that when it hits the right spot will do its little thing like a mousetrap and infect a cell. So, I mean, killing is actually the wrong term probably too. So yeah, inactivate, meaning it no longer is able to infect or becomes non-infectious, meaning it's not going to infect you or kill you.
Great. Thank you for explaining that. Now, here's an interesting question, kind of going back to masks, because that is something a lot of people do use. So we're getting a pretty good amount of questions on that. Now, does it matter what kind of mask is most effective? The fabric type, the number of layers, metallic threads, all of this sort of considerations. Is it just a matter of wearing anything is good, like you said, but is there more effective variants of that?
Yeah, so definitely the makeup of the mask matters. And so just starting on the extremes, so, it's called an N95. And so that means that it filters out 95% of the particles that pass through the mask at a certain size. And so that size is a very small size that would make it to the deep lung. And so that's sort of the most effective thing you can wear. You have to wear it correctly, like I said before. If you wear them incorrectly, that can change things. And then as you start to look at, different homemade masks, they're all effective to some degree. And if you go and start looking, there's a lot of studies being done to look at this now. And some of them, the one extreme is 95% effective. Some of them actually approach that. Others are more like, 10, 20, 30% effective at blocking particles. So what they're looking at is they're going to generate some particles when they do these labs in the test. They're going to put the mask in between, and then they're going to see how many are coming out the other side. And then they're going to remove the mask just to be sure they would all pass through anyway. So that's the sort of scenario you're testing. But even in a case where you have either a poor fit or a poor mask, I mean, if you're blocking 30% of the particles, that's still a 30% decrease in the probability of getting somebody sick. So even, what we would think of as pretty highly inefficient masks, like that we would never wear in the lab from a safety perspective, but that if you made it at home and wore it, I mean, it would provide some protection. So, 30% isn't nothing. So that's a good thing.
Well, thank you for breaking that down. Absolutely. And one thing we can do is that if you think of any of the studies that you can point to, we can always include that on the YouTube video that we'll upload too. So if there's anything you can think of that are a great way to direct them, we can absolutely include that, because I think these are great things to discuss and to consider. Now, the last question, we're all told to wash your hands with soap and water for, a certain amount of time. Now, does that kill the virus? So, of course, we just talked about the terminology. So does that inactivate the virus on the hands? And does it work on surfaces too? Or would you recommend, I've heard things that it's more effective than hand sanitizer, but I'd be interested to hear your take on it.
Yeah, so hand sanitizer works. If you put isopropyl alcohol on the virus for 30 seconds, you can kill the vast majority of it. Soap will inactivate the virus, so it disrupts the membrane around the virus, and it'll make it so it can't infect anymore. So it's also inactivating the virus. The other thing soap is doing is, just physical removal, right? And so I'm washing my hands and I'm rinsing it with water, and so even if it doesn't inactivate it, I'm still removing that virus from my hands, and so that isn't there anymore if I were to touch my face. And so that's an important part that's sometimes not thought about, I think.
Well, great. Thank you for that. And also for discussing the hand sanitizer as well. And so that they're both effective is obviously a relief to a lot of people. Now, I do believe that's it for our questions. Actually, we do have one more. Is an asymptomatic carrier much less likely to infect others? That's an interesting question. We hear a lot about asymptomatic individuals. Are they less likely, or do they still have the same likelihood of infection as, say, someone who does have the symptoms?
Yeah, so that's a really good question. And the answer is we don't know really. And so, like I said, one of the biggest gaps that I think, I mean, in terms of what's remaining that we don't know about this is if I have an infected person, whether they're symptomatic or not, asymptomatic, symptomatic, how much virus are they actually shedding? And when in the disease course are they shedding it, and when does it peak? And so we really need to understand that in order to help understand transmission associated with that. And so that's one of the things we'll hopefully be, trying to tease out in collaboration with some of the folks I mentioned we're working with over the next few months. And I know there's other labs working on that as well, including down at NIH, where they're actually looking at people who come in who are either sick or asymptomatic that have tested positive, and they're looking to see if they're actually shedding virus. And so that work is ongoing. So hopefully we'll have a better answer to that in the next couple of months here.
That's great to hear that, it is something that, of course, that you are looking into, and that hopefully we'll be able to learn more about. This is going to be the last question, and then we'll wind it down. But a lot of great questions today, so we really appreciate everybody writing in and letting us know what their thoughts are and some things they'd like to learn. This is a unique and a great opportunity for a lot of individuals here in Las Vegas and really across the country to pick your brain. So we appreciate that. Now, the last question: can the virus survive in a body of water and then get back in the air as water evaporates, or is that something that you're still kind of learning as you go?
Yeah, we haven't looked at that specifically, but I could imagine that it would survive for some period of time in water. So it depends on where the water is, right? And so sunlight or UV from the sunlight, which is what's inactivating the virus, does penetrate water. So if it was outdoors, it's probably dependent on the environment, just like it is when it's on a surface. But I could imagine it would persist for some period of time. Now, can that be put back into the air as the water evaporates? Not as it evaporates if it's not agitated, but if the water was agitated, and we know this from other diseases in nature, where the bacteria will be in the water, and then as the wind blows, particles can get generated off the water from the waves, and that bacteria will become airborne and people can breathe it in and get sick. So that happens for other diseases. So if it was in water, I mean, it's possible that could happen. But yeah, we haven't looked at water specifically, though.
Well, thank you for taking that last question. That's interesting to hear. That is something that you hope to take more of the look at. So thank you, everybody, for attending. It was a wonderful talk. It was great having a conversation with you, Dr. Dabisch, about something that's on a lot of our guests and people we engage with online. A lot of questions about this. So giving us the opportunity to talk with you and getting it out to as many people as possible, we really appreciate that. So thank you again. And and of course, you mentioned the resources that people can take a look at, and we'll include that on the YouTube video as we upload it. But stay safe out there. Thank you again for joining us. Thank you, everybody at home and across the country watching along with us. And as I mentioned, this will be uploaded to YouTube to watch at any time at the end of this week. So thank you, Dr. Dabisch, and thank you and the team at NBACC for all the great work you're doing to try and keep us safe and informed. So thanks again.
Yeah, thank you very much for the invitation to come share it with you today. This was really fun.
It was fun for us as well. Thank you again, Dr. Dabisch. Bye, everyone.